Hi, my name is Addison Hinson, and today I will be presenting my project on blood typing. This is a topic we learned through lab for my Anatomy and Physiology 2 class, and so I kind of did a presentation on it to help better understand it, and I hope you find this helpful. For my first slide, I will be getting into what blood typing is. Blood typing is a laboratory test used to determine a person's blood group based on the presence or absence of specific antigens on the surface of red blood cells. The main blood groups are A, B, AB, and O. And the test also identifies the Rh factor, which means whether they're positive or negative. For example, like A positive, O negative, which helps ensure the compatibility for blood transfusions and organ transplants. Like when you get blood, they have to know what type you are. Sometimes they prefer blood types over some just because they're needed more. Like O negative is one of the best blood types to have for donating because it can go to any other blood type. So next I will be explaining how we use blood typing in a lab setting through an A&P class. So number one, we understand blood group systems to learn about the ABO and RH blood systems, including how they determine blood compatibility and affect transfusions. Second, we use them to practice laboratory techniques. We gain hands-on experience with techniques like coagulation test, and which is used to identify blood types. Even though this was an online class I took, doing it through lab still really helped a lot. And then studying clinical relevance, exploring the implications of blood typing in clinical scenarios such as blood transfusions, organ transplants, and disease management. Blood typing is a test that identifies your blood group based on specific markers on your red blood cell. There are four main blood types: A, B, AB, and O, determined by the presence or absence of certain proteins called antigens. Additionally, the Rh factor, which we talked about earlier, determines whether it's positive or negative, indicates the important protein on red blood cells. This information is crucial for ensuring safe blood transfusions and understanding how your blood can interact with others. This is just a freshen up from earlier to kind of understand again what blood typing really is and what it's used for. I feel like this was one of the simpler topics and it is really interesting to learn about. Not only in a laboratory setting but another setting like a hospital. This is very important for phlebotomists and nurses who deal with blood transfusions. For the next slide, I'm going to be talking about different blood types. This is kind of why I touched up on the first slide just earlier. That way we can really get into the different types and how they're classified. They are, again, classified by the presence or absence of specific antigens and antibodies in the blood. The main blood types are: first, type A has A antigens on the red blood cell and anti-B antibodies in the plasma. The second type is type B, has B antigens on the red blood cells and anti-A antibodies in the plasma. And then type AB has both A and B antigens on the red blood cells and no anti-A or anti-B antibodies in the plasma. So this is a universal recipient. And then type O has no A or B antigens in the red blood cell and both anti-A and anti-B antibodies in the plasma. So this is a universal donor. So basically, type A can go to anybody. Or can receive any blood type, if that makes sense, and then type O can go to anybody. So when you have these types of blood, they really encourage you to donate. Sometimes they even give you a little extra incentive if you do donate. Additionally, each blood type can either be Rh positive or Rh negative based on the presence or absence of the Rh factor, a specific protein on the red blood cells. Next, I included a visual representation of these blood types. If you look vertically, you see A, B, AB, and O. And then on the horizontal rows, for the first row, we have anti-A. And then for the second row, we have anti-B. So I put these in here just to show you like a visual representation of these blood types. That way you can see them. We use these a lot for activities during the blood typing exercises. And this is a big help. So next I included a long list of instructions on blood typing and using it in a lab setting and collection. So I'm going to go through it very briefly, but first you would obtain a blood sample from the patient using a sterile needle and collection tube. For preparation of the blood sample, you would use the blood to separate plasma and red blood cells. If necessary, place a small amount of the red blood cells on a test plate or slide. Addition of reagents, anti-A reagent, add a few drops of anti-E reagent to the one portion of the blood sample, and anti-B reagent, add a few drops of anti-B reagent to the portion of the blood sample, and then anti-RH, add the anti-RH reagent to the third portion of the blood sample, mixing gently with the reagent and the blood sample on a test plate or slide, and then you would obviously take a observation of the agglutinin, observing for clumping. If clumping occurs in the presence of anti-A reagent, it indicates that anti-A antigens are present. If clumping occurs with anti-B reagent, it indicates that B antigens are observed. And then with the RH factor, if using an RH reagent, observe for clumping to determine if the RH factor is positive or negative. For results, determine the blood type based on presence or absence of clumping, and then clumping with anti-A but not B indicates type A blood. Clumping with anti-B but not anti-A indicates type B blood. Clumping with both anti-A and anti-B indicates type AB, and then not clumping with either indicates type O. RH factor determines the RH factor of the clumping with the anti-RH reagent, and then clumping indicates RH positive, while not clumping indicates RH negative. So you would record the blood type and the RH factor for medical records, and then go from there with your follow-up. Next, I will be talking about real-life applications. This includes examples of situations where blood typing is used, such as in hospitals and emergency situations. So first would be blood transfusions. Before administering a blood transfusion, hospital perform a blood typing test to ensure that the donor and the recipient blood types are compatible. This prevents adverse reactions such as hemolysis, which can occur if incompatible blood is transfused. This is a really important thing, and I know a lot of hospitals take this very seriously, and they have nurses that specialize in simply doing the blood transfusions and the blood typing tests. Next would be for a organ transplant. Blood typing is essential for matching organ donors and recipients to reduce risk of rejection. And then next would be pregnancy. Blood typing is used to monitor the RH compatibility between the mother and the fetus. In emergency situations where blood transfusions are needed urgently, quick blood typing ensures that correct blood type is used. Hospitals maintain a supply of various bloods to address such situations efficiently. Again, this is what blood drives could be used for, or just donating your blood. Paternity testing, blood typing could be used for paternity testing to determine the biological relatives. And then disease diagnostics and research. Blood typing helps with research studies and disease diagnosis by determining disease and conditions may be more prevalent in specific blood types. And then lastly, to talk about the RH factor, it's a protein found on the surface of red blood cells. It is one of the key components in blood typing alongside the ABO system. RH positive, if the RH factor protein is present on the blood cell, the blood type is RH positive. This means that the red blood cell can have an RH antigen. If it is RH negative, the RH factor protein is absent from the red blood cell and the blood type is RH negative. This means that red blood cells lack the RH antigen. And the role of this in blood typing is crucial for determining the compatibility in blood transfusions and organ transplants. Thank you for watching my presentation. I hope you found it helpful.